extensive work on aflatoxin epidemiology, particularly in the sub-Saharan country. Michael is well known in the field. He is also the Toxicity Committee member of Food Standard Agency of the United Kingdom government, and so many other committees as well. So welcome, Mike, here. It's good to see you here. Yeah, thank you.
Okay, good morning everybody. It's a great pleasure to be here on my first trip to India, so I'd like to thank the organisers very much for giving me the opportunity to come and speak to you. So I'm going to be talking about aflatoxin exposure, particularly in sub-Saharan Africa, but as you'll see, this includes some Southern Asian populations. So the title, you can see mortality and metabolomic changes associated with aflatoxin exposure in an acutely ill cohort of children. And this is part of the chain cohort. And the work that I'm talking about, although the consortium infection and so on, but there is also acute aflatoxicosis, which is kind of severe poisoning. So in countries in sub-Saharan Africa, often where people are growing their own food, storing their own crops, and maybe in the non-ideal conditions, when it's very warm and humid, this promotes growth of the fungus that produces the toxin. So you get high exposure where there are high levels of contamination in the food, but also where people are eating a lot of the same type of food on a regular basis. So in East Africa, maize is one of the staple diets, and that's very prone to contamination. And in West Africa, peanuts, as shown in the top there, they're also very prone to contamination. So aflatoxin, as I said, is a human liver carcinogen. It's established, very well established, the mechanism. You get metabolism to a reactive epoxide in the liver. That reactive dial epoxide binds to guanine in DNA to give you adducts that are mutagenic, and you get a very well-known pattern of mutations associated with liver cancers in areas that are high in aflatoxin. And because aflatoxin reacts not just with DNA, but also with proteins, there are a number of different biomarkers that can be used to study aflatoxin exposure. Clearly, if you're interested in cancer, DNA adducts are the most relevant, and many of the early studies used DNA adducts, and also metabolites of aflatoxin in either urine or plasma. But we are using the albumin adduct. So the same dial epoxide that causes DNA damage also causes binding to proteins, and albumin in the blood is a good source of a protein that binds aflatoxin, so we can look at levels of aflatoxin adducts to albumin as a measure of exposure taken from blood samples. This is a bit hit and miss. And this is the method that we've been using, or not just me, but other people at Leeds before me have been using for kind of over 20 years. Originally derived by Chris Wilde when he was working in IARC in the early 90s, where they developed an antibody against aflatoxin and then used that to develop an ELISA. And we are basically using the same ELISA method. It's semi-quantitative, it's got a good limit of detection, 3 picograms of adduct per milligram of albumin, and it's being used now in lots and lots of studies, and it's been validated against dietary exposure. This was done in the 90s by Chris Wilde in adults in Africa, but in 2014 we published a study for child exposure in Tanzania, where we can see that there is an association between dietary levels and the biomarker. It's obviously not perfect, we've got a lot of variation, Some of that may be because the measures of the dietary intake are not very accurate, which is one reason why it's better to have a biomarker. And of course, you also get individual variation between how the aflatoxin is metabolized. There's been a lot of studies, particularly in sub-Saharan Africa. We published a review of some of these studies back in 2018, and you can see highlighted several countries where we've had populations we've been able to measure the biomarker. And you can see in the graph there we've got levels of the biomarker from a number of different studies. And you can see this is generally levels around 20 to 50 picograms of aflatoxin per milligram of albumin in these countries, sometimes higher, sometimes lower. And of course, these are all snapshots. So you take samples at a particular time point and you measure the exposure. That may vary from year to year, depending how much aflatoxin was present in the crops in different years. It varies from season to season, because as the food is stored after harvest, there is a build-up of the aflatoxin. So if we went back to one of these populations in a different year, we might get a different result. If we were to take samples from people in the UK, we would probably not detect any, because in countries like the UK, all the food is being tested, the likely crops are being tested. For Africa, we see around 50 picograms per meg. These results are showing tremendously high levels. So if you look at the chart here on the right-hand side, it goes up to 20,000 to 32,000 picograms per meg. Hugely inflated. And these are samples that came from an outbreak of aflatoxicosis in Tanzania in 2016. So this was an incident where people ate high quantities of aflatoxin-contaminated food in a particular region, and there was a number of people who became ill, and 20 people died. And at the time, it wasn't entirely clear if it was due to aflatoxin. So the WHO sent us some samples to analyse, and we were able to confirm these high levels of aflatoxin adults in the cases. There were some people here from the same region who were not sick, but also had high levels of aflatoxin adults. On average, not as high as the cases, but still much higher than normal in some cases. And again, highlighting that the effect of the aflatoxin will vary from person to person depending on a number of factors. So we've had a number of studies in sub-Saharan Africa where we've looked at effects on child growth. I won't go into these today, but we've seen an association between impaired growth in children under 2 years old and levels of aflatoxin exposure. But I'm going to talk today about some results from this train consortium. So this is a large consortium that was set up by people at the University of Washington and in America and Oxford University in the UK to look at factors that might influence the risk of mortality in severely ill children who have been admitted to hospital. So these are cases of children who are so ill, often due to malnutrition, that they are admitted to hospital. And it's been found that it's difficult to predict just on clinical symptoms which children are most at risk of dying, although there is clearly a high risk among such children. So this study was set up to explore that. This was the actual protocol that was published. You can see a lot of names here because it involves a large number of different study sites. As you can see here, so there are a number of study sites in Africa where samples were taken, and also some in Bangladesh and one in Pakistan. So the idea here was that as the children were admitted to hospital, they would be recruited through getting the consent of the parents or caregivers into the study, So samples were taken alongside the normal sort of blood samples that would be taken from these children as part of their treatment. And then they were followed after discharge for a period of about, I think it was about 6 months, to see what sort of factors might influence their risk of death. Next slide. And alongside simply studying kind of the various factors that might influence this, there were also a lot of laboratory studies undertaken to try and investigate possible mechanistic reasons. So you can see there's all sorts of different omics technologies have been applied by different laboratories, proteomics, polysaccharides, metabolomics, etc. Down the bottom there you see University of Leeds, and we did the serum aflatoxin. So we were brought in to ask the question whether aflatoxin might be associated with any of these risks. So as well as the 3,000 children recruited into the whole study, there was a nested case cohort set up, so with about 1,200 children from whom the biological samples were taken for the various lab studies. This included a random selection, and then any remaining cases, so the children who had died were then added in as well, and there were also community participants where samples were taken at the same time as the children were admitted to hospital to look at the background levels in that community. And then, as I said, samples were taken at admission, and then at least a kind of a balance between cases and controls for most of these particular potential differences, although you will see there are some slight differences there. So, for example, breastfeeding slightly higher amongst the survivors, which might be a relevant factor. And we've got various other factors. This shows the aflatoxin exposure characteristics. So we have about half of the samples that were measured, and not all samples it was possible to get a measure from. We were the last ones to get hold of the actual samples, so in some cases there wasn't sufficient left. But in the ones that were measured, about half were below the limit of detection, and about half above the limit of detection. And then at the bottom there you can see that the median for the levels was 8 micrograms per milligram. So this is kind of what we often see in sub-Saharan Africa. That's the median of the geometric mean. Next slide. And then this shows the levels in the different populations. So we have the different sites here. And the key finding here is that the levels, kind of as we expected to see, are higher in the African populations than in the Southern Asian populations. This is something that we've seen in previous studies. There's variation between the different sites, with Kenya and Uganda, Malawi having higher levels of exposure, which again is something that we've seen in some of our previous studies. And in general, you've got the community participants at the bottom there, and we can see that among the community participants there is still reasonably high exposure. So it's nothing specific about being exposed and then being ill and being in hospital, because these populations, a lot of the people are exposed to these chronic levels of aflatoxin. We saw a reduction in levels between admission and discharge, which is what you would expect, because of course the children are getting a different diet in a hospital. So one of the things to mention here is that the aflatoxin albumin biomarker kind of integrates exposure over the previous couple of months because of the lifespan of albumin in the blood. So it's not measuring exactly very recent exposure, but nevertheless, over the period they were in hospital, on average, we saw a decrease. As we've seen in other studies, we get an increase in exposure over age. Once the children are weaned and start to eat family food, of course their exposure increases, and by about 2 years of age they're getting the same sort of levels of aflatoxin exposure that we would see in adults, as shown on the right hand side there. there was a correlation between the biomarker levels and socioeconomic status, again something that's been observed in the past, and this is explained because people who have slightly more resources, and you know, none of these communities are well off, but people are able to look at whether there were any candidates metabolites from the metabolomics analysis associated with aflatoxin exposure. So we were able to get hold of the database for metabolomics and then analyse it in terms of associated with aflatoxin. I won't go through the methodology because I'm not an expert and I'm short of time.
It's your first time in India, so I would like to welcome from the land of diverse culture. So my question is regarding if you have mentioned about the ELISA test, and there are several other tests also available like chromatography techniques like a TLC, HPLC. Which one is the better one, sir, for the prevention of aflatoxin? And if we can hydrolyze into metabolic activity, it should be converted into the AF-M1 variety. So which one is at that time which should be caused aflatoxin effect in a milk? I mean to say which test is better, effective, like ELISA or chromatography technique, sir?
Right, so I'll try and quickly answer that. So the other method that is now more widely used, I would say, is HPLC with mass spectrometry. So MSMS is now able to handle more samples and is very sensitive. So a lot of labs are using mass spec to look at the lysine adduct, which gives you a much more specific measure, but also showing kind of similar results in terms of aflatoxin exposure in the population. You talk about aflatoxin M1, yeah, you're quite right, that's a metabolite that is formed from hydroxylation, is present in milk, so even breastfed children are exposed if their mothers have been eating aflatoxin contaminated food, aflatoxin M1 in milk. You can also get M1 in the milk of cattle that have been exposed to aflatoxin. There are kits, ELISA kits for measuring M1, which I think are fairly reliable, but all of these things have to be optimized if you're using them. I missed the first question that you asked because somebody coughed, so I don't know.
You can discuss.
Please talk to me later. I'm here always.
So grab him and ask questions. Yeah.